Densification of the fascia is a bit of a hot topic at the moment, and so I thought I would put together this lesson that summarizes for you the best available current evidence about what they are and then what we can do about these events in our tissues. Now, I'm coming at this through a lens of manual therapy and movement, being the two lenses through which I approach most of these types of things. Now, you might be coming to this as part of the fascia series called The Truth About Fascia that I've put together, in which we tackle a series of other topics related to the fascia. Or if you're coming to this separate to that, know that below this video somewhere is going to be a link to the registration page for the Truth About Fascia series, in case after taking this lesson you are interested in learning more about the fascial tissues and going through and breaking down a series of commonly perpetuated myths that we encounter out there. Okay, you'll get a bit of a taste for how I break things down here in this video. So hopefully by the end of it, you'll get an idea as to whether or not you'd like to dive into the Truth About Fascia webinar series with me. So we're going to talk about densification. We're going to talk about what they are. We're going to talk about the physiology of these densifications, what we currently know about that, and then what we can do about them through the lenses of manual therapy and movement practice. But first, before we do that, why is it important? Why do we care about densification? And why do we care about all of these types of things as it relates to the body? Well, number one, and this is a pretty important one, they contribute to the burden of pain for people. All of us at some point, and probably at many points across our lifespan, will experience pain and pain syndromes. And this is one of the puzzle pieces in understanding more about sensation and what we might be able to do about it if we're encountering that ourselves, or if we work with other people who might be experiencing some of these things. And when we know more about these things, we can do more about them, and we can have better outcomes in a more reliable fashion when we understand what the science is telling us about these types of things. One thing that I want to be really clear on is that densifications in our fascial tissues are one component of the broader image on pain. Okay, we are going to be zooming in on that component, but at the end, I'll also remind you that it is just one element of the pain puzzle, essentially. What we can also do with this information is we can target our work a little bit more specifically. We can tailor, if we're able to identify that someone has some densification of their tissues, we will be able to target that a little bit more specifically. And then also what this does, interestingly, and I'll mention this more at the end as well, is it provides a potential reason for some of the therapies that we aren't necessarily able to produce really reliable benefit from, or we aren't able to reproduce benefits from in any sort of consistent fashion. This might explain why some of these things work for some people. Okay, so I'll go through that in a little bit more detail at the end. And essentially, when we have more knowledge, especially about the things that contribute to the pain puzzle, we have a bit more of an integrative view of the body, and also a bit more of an integrative view of pain as a whole as well. So that's the context for why it's important to go through and study these types of things. So first up, what is the physiology? What is happening here in the tissues that causes densifications? Before I actually mention this, I need to table something and be very honest with you and say that when I first heard the term densification, I thought it was one of those social media terms that was basically made up to describe something that we experience as manual therapists, because it sounds like one of those cheap substitutes for an actual thing, right? Little did I know that going in and doing a bit of reading and research actually sort of knocked me down a bit and made me understand that it is a term that is being used in the literature, and actually more and more we're starting to see it show up, and we're starting to see more and more papers and research being done on the topic, which I think is exciting. So with that out of the way, what is the physiology of densification. We've all heard various stories about the fascial tissues, and again, in the Truth About Fascia series, I go through and break down a lot of those myths that we hear. And specifically, as it relates to densifications, we're talking about an increased concentration of what's called hyaluronic acid, or which is abbreviated to, in papers and here throughout the presentation, it'll be abbreviated to HA, hyaluronic acid. Why does this happen? Why does hyaluronic acid accumulate? Well, one of the reasons that we've been able to identify so far is inflammation. Okay, inflammation is often accompanying our accumulations of hyaluronic acid, and so we know that there's some degree of linkage there. The questions around that might continue to evolve. We might, for example, be able to determine whether or not the accumulation of hyaluronic acid is a symptom of or a driver of inflammation. That might be an important thing that we learn in future research, but it's not something that we currently understand from what's available in the research. We've also seen that stagnation or stillness is something that also might contribute to accumulation of hyaluronic acid. What happens with these accumulations of hyaluronic acid? Also, by the way, again, if hyaluronic acid is a new term for you, I think it's important that you dive into the Truth About Fascia series, because there's a whole lesson where we talk about hydration of the fascia, and I'll mention more about that shortly as well. But within that lesson, hyaluronic acid features heavily, and I go into a lot more detail on it there. So for now, I'm going to sort of skip over or just briefly brush what hyaluronic acid is. And if you want to know more, find the link below for the fascia webinar series, The Truth About Fascia, where I will absolutely take you through what hyaluronic acid is in a much more detailed fashion. So essentially, hyaluronic acid is one of our glycosaminoglycans that we find in the ground substance of our fascial tissues. Very briefly as well, our fascial tissues, we have a fibrous component and we have this sort of liquid gel-like component, which is the ground substance. Oftentimes, when we talk about the fascia, we focus heavily on, and I will say that I do that a lot as well. We focus heavily on the fibrous component of the fascial tissues, being collagen, essentially, with a bunch of other things. And though this lesson is mostly going to be focused on the ground substance, because that is where hyaluronic acid is found. Within the ground substance, we have glycosaminoglycans, which hyaluronic acid is one of them. We have glycoproteins, and we have a whole bunch of other things going on within the ground substance as well. When hyaluronic acid accumulates, it builds up in a very focal area, in a specific spot, and normally hyaluronic acid draws with it a lot of water, huge volumes of water actually. And though, for whatever reason, whether it's lack of movement or inflammation or some other reason, this hyaluronic acid is coming together, and in preference to, or instead of drawing water with it, it starts to create cross-links between itself or between other molecules of hyaluronic acid, between proteoglycans within the ground substance, and even between our collagen fibers within our fascial tissues. And what ends up happening is the formation of what's called supramolecular structures, which is basically a big word to describe big molecules that are forming from other molecules coming together and having an interaction. With those supramolecular structures, I'll also be clear in saying that the bonds between them are relatively weak bonds. They're not bonds that you would equate to the bonding that we see between collagen fibers within the fascial tissues. This is something separate to that. And we would call this group of weaker interactions van der Waals forces. Okay, so what happens is hyaluronic acid starts to interact with other molecules of hyaluronic acid, proteoglycans, and even with collagen fibers to form this sort of matrix within the ground substance. And you end up seeing an increase in viscosity in that area where the accumulation is present. What happens as a result of that increased viscosity is you get less glide of the tissues, which in certain formats, part of the purpose of hyaluronic acid in our body is to create more lubrication and more glide. Now, as a sort of a detour here, I will also mention that there's many, many functions of hyaluronic acid within the body, and it is by no means limited to lubrication of tissue movement. And in certain formats, in certain parts of our body, that is one of the things that it participates in. And so when we see hyaluronic acid, instead of in binding with or interacting with water, where it normally would do that in our fascial tissues, we see it interacting with other hyaluronic acid molecules, proteoglycans, and collagen. We see that the viscosity increases, and therefore we get less glide. Now, something else important here on this slide that I want to really highlight. This diagram is an image generated, I imagine, by AI. And it does a couple of things. It does a couple of things well, and it does one thing poorly. The poorly piece is the dehydrated. I've crossed that out, and I'm going to take you through that shortly in a couple of slides' time. But I'm going to suggest to you that we don't think of densifications as dehydrated tissue, and I'll explain to you why. But in the same vein, this image has done something quite well in showing this pooling of liquid being, we can assume, water within the fascial tissues. And that's important. And I'll show you why in just a moment. It's important because part of the physiology of our densifications is that whilst we see within the area of hyaluronic acid accumulation, we see that there's less capacity for hyaluronan to bind with and interact with water. That water still stays there in the fascia. And this interesting study, which also, by the way, another side little note, is that all of the studies that I've read that have contributed to the formation of this lesson, I've saved in a file, and you have access to that file. I'll put the link below this video as well, so that you can go through and actually read the studies if you would like to do that. And number one, check the receipts, basically, but also two, if something like this interests you, you can dig into a little bit more detail on the matter. But basically, this really interesting imaging technique, which is relatively new. It's an MRI, but it's not the normal MRI. It's a row T1 type MRI. And what it does is it helps to visualize the freely available or the unbound water within the tissues. And you may not be able to see it all that well on this magnification, but I'm going to bring the cursor in just to show you. In the series of images in the pre-manual therapy imaging, you'll see where the arrow is pointing. You'll see that there's red. There's more red within the surface layers of the tissue, okay, than there is on the post-manual therapy images. You'll see that there's basically no red in all of these posts. There's maybe a little bits here and there. That red is a demonstration of unbound water. It is the representation of unbound water in the fascial tissues. And so this is why it's important to reframe our idea around these densifications being dehydration of the tissue, because the water is there. It's available. It's just unbound, where we would regularly see it bound with our hyaluronic acid or interacting with our hyaluronic acid. As a result of the accumulation of HA, we have seen that the water is excluded. However, it's not gone from the tissue. And so because of that, we shouldn't really be saying that it's dehydration of the tissue. And I'll give you an example of what I mean about that in the following slide. But I also wanted to show you what it looks like on a special type of ultrasound as well. These are also densifications that we've been able to image on ultrasound. And in particular, we're looking at the section that is the brightest white on the image and also with a red line through it. The red line is basically a measurement of how thick that densification of fascial tissue is, or how thick the fascial tissue is. And we can see that in our pre-manual therapy image, we can see that it is thicker than in our post-manual therapy image, suggesting that as a result of the intervention being manual therapy, we've been able to reduce the thickness of our densification. And why is that important? It's important because for females in particular in this study, and I think it's important that future study involves more people because the number of people in this particular study wasn't great. But essentially, in females within this study, we were able to determine that thickness of a densification is correlated with intensity of pain related to the densification. For some reason, in males, that association was not seen. Now, that could be as a result of many things. It could have been simply as a result of not enough people in the study. Oftentimes, we don't get to see the full picture of those patterns until there's a big number of people in the study and we can draw proper conclusions from that. But it could also be related to other things. And this is me speculating, but it could be related to the things like we would typically suggest that females tend to be a little bit more in tune with their bodies. As a result of all of the healthful things that people participate in, usually speaking, females are the ones that have a bit more interest in doing those things for themselves. And as a result, they pay closer attention to their body and how things respond in their body to what they're doing. As a result of that, they might be able to distinguish a little bit more accurately how these densifications contribute to sensation and might be able to place them on a graduation of more or less intense. And we've seen then that the intensity score that they give their densifications is correlated positively with the thickness of the fascial tissue in the area. I just want to mention, no, that was actually all I wanted to mention on that slide. I've got some notes just to make sure I don't miss anything. But essentially, we see that the thickness is important for some people. So let's just backtrack for a second. Wait, so it's not dehydrated. I'm going to say that no, it is not dehydrated. And I say that I'm going to say it because I could also see how some people might suggest that it's dehydration within a densification. And though, hopefully with the analogy that I'm about to give you, you'll see why I don't think we should be calling it dehydration. Number one, there's a whole lot of other science that suggests to us that dehydration of the fascia, while we are healthy and not in the hospital in a very serious critical condition, is probably not possible, and if it is possible, it's highly unlikely to occur for most people. We are more likely to see something like the fascia dehydration in a scenario where we are clinically dehydrated and in the hospital, okay? In that situation, it's probably going to be occurring. However, before that, if we're talking about preclinical or non-clinically relevant dehydration, it probably doesn't happen. And I go into great detail of why that's the case and the biochemistry behind that in the Truth About Fascia series. So if you want to know more about that and those interactions, I recommend taking that series so that you can get that download, basically. And this is important to note. I will sort of also say this is important to note because a lot of people are out here suggesting that our densifications are dehydration. And even in the image that you saw before, it is talked about as a dehydration. And I think it's important to step out of that because there is enough water. The water is present in the fascial tissues. And dehydration is a lack of water available. But in this situation, in our densifications, there isn't a lack of water available in the tissues. What has happened is, for whatever reason, it is unbound. So it's freely available water. And in my books, that does not mean that there is dehydration present within the tissue. If this unbinding of water caused that freely available water to be reabsorbed, maybe I would concede that that could be in the realms of dehydration. But that's also not the case. The water stays there in the tissues, and you'll understand why that after the Truth About Fascia series. But essentially, when water is available, it's just a matter of utilization or distribution of that water within the same tissue. It's like, and this is the analogy that I'd like to provide you, it's like if we were to image someone's digestive tract, okay, and if we find a spot within their digestive tract that does not have food in it, will we make the assumption that they don't have enough food in their digestive tract simply because there's no food in this one small portion of their digestive tract? What if we were to then zoom out and see that, well, there's food after it being digested, and actually they've half an hour ago eaten a meal, so there's also food on the way, right? There's readily available food. There's more than enough food available within the digestive tract as a whole. And so we can't suggest that in that instance there's not enough food, right? It's the same if we apply that logic to the tissue here, the fascial tissue in particular. The water is available. It's there. It's in the system. It's in the tissues. It's just not bound to hyaluronic acid like it normally is. So it's a distribution issue, not an actual volume issue of water. And I think that's an important distinction to make, because that helps us to step out of this idea that it is dehydration and instead recognize that it's more about this accumulation of hyaluronic acid excluding water from a very small portion of our fascial tissues, but the water stays there and is readily available to be rebound if we're able to disrupt the interaction that hyaluronic acid is having with other hyaluronic acid molecules, with proteoglycans, and with the collagen fibers of the area. All right. Now, let me just see if there's anything else I wanted to mention there. No, I think that is it. Here's the next step. How do these densifications lead to pain? Because it's all very well and good for us to understand the physiology behind them, but what does that cause? The hypothesis is, and I emphasize that word because we have not definitively proven this yet, the idea is that with a decreased glide of the tissues, it's hypothesized that this irritates local nociceptors. And our nociceptors are the receptors in our bodies that detect noxious stimuli, and typically are associated then with a pain response signal from the central nervous system. That is one possible way in which our densifications can lead to pain. Again, this is not proven. I'd also like to highlight that pain is complex and oftentimes coming from many directions. And another sort of idea that I'd like to plant in here is that even if the increased viscosity of the tissue does not lead to an increased irritation of our nociceptors, it could be the associated inflammation that comes along with our densifications that does the same thing. We know that certain inflammatory markers tend to irritate or trigger a response from nociceptors, and so it could be, instead of our viscosity of the tissue, it could be that the inflammatory markers elicit this pain response. So it could be the viscosity, it could be the inflammation, or it could be a combination of those two things. Or, again, because the research is emerging in this area and isn't very extensive, it could be something else that we have not yet measured or seen. And what we have seen, so regardless of exactly what is causing the pain or leading to our pain signaling as a result of densification, we have seen that it is associated with pain. We see that densifications lead to pain because when we are able to decrease the densification or remove the densification, we see a decrease in sensation and pain. So therefore, we see that they lead to or contribute to part of the pain puzzle that people are often experiencing. I wanted to also take a moment within this to distinguish densifications from trigger points. If I've seen you in other courses and in other parts of the world, I do spend a lot of time talking about trigger points. And in some ways, densifications seem similar to trigger points. However, I would like to sort of create a bit of a distinction here. Trigger points are specifically muscular in nature, so they occur within muscle fibers. And I also have a separate mini course on the trigger points if you'd like to know more about that. I can also put that link down below if you'd like to explore that a little bit more. But trigger points are uniquely muscle based. Densifications are uniquely fascial tissue based. And so when we are exploring the body with our movement practices or with our manual work, we have to be able to distinguish between what's fascial in nature and what is muscular in nature in an effort to be more effective in the work that we're doing. So they are distinct physiological events. And though they do seem to, at least with what the research is currently proposing around densifications, they do seem to have similar pain profiles, and they also seem to participate similarly in pain referral or sensation referral, which is the idea that, or is the event where a trigger point or a densification in one region leads to sensational pain in an area far away from that point. Okay. And I think that that is important for us to know so that we're able to provide different focused treatment options based upon what we are experiencing in our own bodies or with the people with whom we work. By the way, if you do want to really understand more about the trigger points, then I'll put that link below so that you have access to that. One of the key things about our approach to trigger points is static ischemic pressure. Basically, generous pressure that is held for a significant amount of time until we experience a decrease in intensity or a decrease in the density of that tissue. Because also, in terms of our manual approach, if we're looking through a manual lens right now, trigger points also feel like a dense area, dense focal point within the tissue. We have to get good at, as manual therapists, distinguishing between whether that dense focal point is in a muscle or whether that dense focal point is in the fascial tissues. So here's what we can do about densifications specifically. Now we're coming back, we're leaving trigger points behind for now, and we're coming back to our densifications. What can we do about them? Well, so far, and I'll highlight again, so far what we've seen is that manual therapy is able to help. There's a small amount of research that has been done on this. The technique is interesting because in a lot of our other fascial related work, and again, I mentioned this a moment ago and I'll reiterate this now, in a lot of our other fascial work, we're very focused on the fibrous component of our fascial tissues. And I will be the first to put my hand up and say that in most instances, that's what I talk about. When I'm referring to fascial release, I'm mostly referring to the fibrous element of our fascial tissues. What I will do a better job at in the future and moving forward is distinguishing between the ground substance of our fascial tissues and working with that and working with the fibrous component of our fascial tissues because they require distinct approaches, seemingly. Because what we've seen with the handful of studies that have been done on the release of our densifications is that we've seen an application of what's been termed fascial manipulation. So in the research, it will often be abbreviated to FM, okay? So you might start to see that showing up as well. And that is a very different manual technique than what we would experience in something like a rolfing treatment or a treatment that is otherwise focused on fascial stretch, let's say, for example. We're thinking about, or we're looking at our fascial manipulations being medium to low amounts of pressure and held for only 30 to 45 seconds and using oscillation. You can actually see an application of that in one of the studies here on this image. They imaged the technique that they used, basically a medium to light amount of pressure over that densification. And along with that pressure, we see some movement. Whereas with other fascial, again, with the fibrous component of the fascia, we would be looking at static application of shear force. We're taking things in different directions and we're holding it in order to have an influence. But for this is quite different. Small amount of pressure and oscillating instead of maintaining it still. The theory is behind this application, and again, I say theory and I highlight that because we have not been able to definitively demonstrate that this is exactly how it works. However, the idea is that by providing this small amount of pressure and this oscillating force, we're able to free up the binding sites of hyaluronic acid, which allows water to rebind or reinteract with our hyaluronic acid, and that returns the function of our hyaluronic acid to one of lubrication of the tissues, allowing that viscosity of the tissue to drop and allowing an increased glide of those tissues past one another, ultimately leading to an alleviation of symptoms. Okay, so manual therapy is one of the things that we've seen so far that helps. Another thing that we've seen that helps is hyaluronidase injections. Hyaluronidase is the enzyme that basically breaks down hyaluronic acid into smaller chunks. We have seen that when those injections are guided by ultrasound, and a couple of the studies within your resources folder demonstrate that, when ultrasound guided application of hyaluronidase has been done, we have seen a resolution of the densification and therefore a resolution of the symptoms as well. We've also seen the same type of results with what's called hydrodissection, which is similar to the application of the hyaluronidase injections, except it's just a saline solution injected into the same spot. For me, and this is a question I'll bring up later on, that is sort of a rhetorical question that I hope researchers start to explore as time goes on, but it's essentially, what is the difference then between the application of an enzyme and the application of saline? And why do they both work? Do they both work to the same degree? Is one more effective than the other? There are lots of questions that I have surrounding that we don't yet have answers to. One of the proposed ideas is that both hyaluronidase injections and hydrodissection have an effect, or at least have a partial effect, due to the pressure of the injection itself. When you're injecting a liquid into your tissues, and you've probably experienced this if you've had any type of injection, you'll notice that where the fluid is being added to your tissues, there's an increase in pressure. Pressure builds up. And so with our hydrodissection specifically, the hypothesis is that because of that pressure, it is enough to disrupt the bonds or the relationship between hyaluronic acid chains and within our densification, and thus restore the interaction with water. The same is true then for the hyaluronidase. The hyaluronidase breaks down those interactions because it breaks down the hyaluronic acid chains into smaller chunks, which then allows those interactions to occur with water again. So we've got manual therapy, and we've got very specifically ultrasound guided injection of saline and hyaluronidase as being our options. We've also had the proposition that heat is part of a recipe that can help with our densifications. Some are suggesting that that's one of the elements of our manual therapy application. However, that has not been directly measured yet. And we also do know that in some instances, in a lot of instances, sustained manual therapy does alter the local temperature of the tissue. The question that I have around that is whether or not 30 to 45 seconds of light or medium pressure with oscillating application is enough to elicit a temperature change in the area. We don't yet know that, basically. So the idea is that yes, heat, or the theory is that yes, heat will help. And we have seen that in the application of sauna and general muscle stiffness. And so I would also like to highlight that that is not specifically showing us that it works in the same capacity as our manual therapy and our injections. All right. I want to be clear in saying that that is a hypothesized or a proposed mechanism that works when it has been looked at on a much broader level, so on a much higher level than what we are trying to focus in on. Okay. So it hasn't yet been measured directly, and the assumption is that increasing the temperature will assist with the degradation of these interactions in a densification. Now, why is it important that we do something? Why not just let the body do its thing? In a similar way to with trigger points, how they can become persistent, how they can contribute to substantial amounts of pain and discomfort, and they can be really persistent and difficult to get rid of. In a similar way, our densifications can lead to quite substantial dysfunction and disorder if they are allowed to continue. One of the ideas that is gaining steam, and we can see mechanisms behind, however, again, it's not definitively shown, is that densification is a primary phase or a primary step in the process of fibrosis. And fibrosis is a much more medically significant circumstance. You might know examples of fibrosis like frozen shoulder, for example. There are many examples of what fibrosis can cause, but frozen shoulder is one example. And when we have frozen shoulder, the impact and burden on our health is quite significant. And for a lot of people who have fibrotic conditions, the recovery is limited. I don't want to say that that is going to be the case for everyone, because there's a lot of ingredients in the recipe of rehabilitation from fibrotic events. And what I will say definitively is that fibrosis is much, much harder to manage, especially without surgery, if you want to go without surgery, than densifications. And so it's much better for us if we're able to get on top of things while it's still a densification than if we allow it to continue and for it to develop into fibrotic tissue, which oftentimes requires a combination of surgical intervention, significant manual and physical therapy, and over a much longer period of time. So essentially, unmanaged densification can develop into fibrosis. What are the things that we know don't work? One in particular is pH changes. And I am specifically highlighting this because, one, we see that pH is implicated in a lot of videos out there in the social media world on the formation and dissolution of densifications. And we know, though, based on the current available evidence, that it's not a factor. And what I will also say is that there is a paper within the folder of research that I've read to build this. There is a paper that suggests that it is part of why our densifications form. But this is where it's interesting. This is where research gets tricky, because if you then follow the reference for that statement, the reference for that statement does not, in fact, say at all that pH is an influencing factor. It is that has been poorly interpreted by the writers of the paper and misused, misrepresented, basically. What we can see is outside of physiological conditions, pH does have an influence on how hyaluronic acid behaves. However, our body is very good at maintaining a pH range within a very small set of numbers. And so within that range, what we call the physiological range of our pH, we see that there is no change to the behavior of hyaluronic acid within a physiological range. All right. So we want to take pH off the list. This also means that if people are out there suggesting that drinking specific types of mineral water or alkaline water or this, that, and the other special type of water, suggesting that that will help to reduce your densifications, that's a no. That's not how it works. For multiple reasons, but it's not something you can basically just ignore that. We know that pH within a physiological range in the tissues has no effect on how our densifications occur or how they're resolved. The other thing that I've put here is just a series of question marks, because the rest of it we don't yet know about. There are bound to be other things, other claims that will be made about the release of our densifications or avoiding densifications. And there's bound to be some things that are false currently. I've got some question marks there because we don't yet have a list of those things. Some extra thoughts that I'll leave you with as we start to wrap up this lesson. Some extra thoughts that I think do a couple of things. I think highlight where we're at with our research on densifications. It's still relatively new as part of the umbrella of fascia research. It's a small component and it's relatively new. And with what we understand currently, with how manual therapy and specific forms of manual therapy have an influence, with our hyaluronidase and our hydrodissection applications and heat, how it's all proposed to have an influence on the release of these densifications. This might, an emphasis on the might, this might provide reasoning or might give us an understanding as to why certain things like cupping, and that's why I've included the image of cupping here, why cupping can be really significant for some people, yet on a broader scale within that avenue of research as it relates to cupping, we've not yet been able to produce results that really demonstrably show that cupping is effective and important for a lot of people. Which I think is exciting, because what it means is that there are still elements of our physiology that can be influenced by these practices, which can ultimately be, if they are distributed and applied in appropriate situations, that might help us to understand how we can extract the most benefit from a practice like cupping, where we've maybe historically been using it too broadly, for example, to reliably show its benefit. So I think this research on densifications takes us down an avenue of potential of, maybe some of these practices that Roger says don't work for him, but Betty says do, right? Maybe this is why some people experience benefit from it and others don't. Okay. What I would also like to see, an extra thought on some things that I would like to see out there, and thus we don't yet have the available evidence to be able to create a scenario with, is I would love to see a comparison of the injection of hyaluronidase and our hydrodissection. I would love to see what elements of those injections are actually producing results. And then, what that might mean for the application of manual technique. Because if it's a mostly pressure-related situation, well, we might be able to entirely reproduce that with our manual technique. as long as the fascial tissues are readily available with, how we apply manual therapy in a normal context. I would also love to know how much pressure is required, both in terms of our injections and in terms of our manual therapy. Is there a limit, right? Is there a certain amount of softness that is too soft? Is there a certain amount of intensity that's too much in order to have a releasing effect on a densification? And then I would also love to sort of be able to help you better distinguish between trigger points and densifications. As of this present moment, my only suggestion is that know your muscular anatomy so that you are better able to identify when you are working with the trigger point and when you are not. And if you are not working in skeletal muscle, then it could be possible that you're at that point working with a densification. And so you would utilize different techniques to approach those different scenarios. So, a brief little summary to wrap this up. Number one, they are real and the term is not as bogus as I once believed, right? That is, again, I'm putting myself out there because I do sometimes react quite strongly to seeing misinformation out there. And in this particular instance, I was pleasantly surprised to know that my reaction was ill-advised. Densifications produce and contribute to the burden of pain, and that is on an individual level and on a much broader level as well. The physiology of our densifications includes aggregated hyaluronic acid into supramolecular structures, and I've spelt it incorrectly there on that side, supramolecular structures, which end up leading to an increased viscosity of the tissue, or that particular portion of the tissue, and a decreased glide in that portion of the tissue, which is often also associated with inflammation, and either as a result of one or the other or both of those things, we see that our local nociceptors are triggered, eliciting a painful response. We see that manual pressure, injection of hyaluronidase, and hydrodissection, as well as a proposal for heat, all appear to work in dissolving or releasing our densifications. They are not trigger points, they are distinct from trigger points, and yet they have a similar pain profile, and they also both appear to refer sensation to other areas of the body. And the last little note here is that lactic acid doesn't make them happen, and that is a throwback to the pH piece, where there has been a suggestion out there that lactic acid buildup in the tissues leads to these densifications. Again, that is not the case based on our physiological range of pH within our tissues. And here is a series of links to the images that I used in the slides. If there isn't a link to the image, it means that the image came from one of the published studies, which you do have access to in the folder for all of the readings related to this lesson. If you have questions about densification, please reach out. If you have even some comments that maybe some of the things that might have surprised you in this lesson, I would love to hear about it. If this sparks an interest to learn more about the fascial tissues through this type of lens, through an analysis of the current best available research, then I really strongly encourage you to dive into the Truth About Fascia series. Again, the link is below. I know I've been pushing that, but I also really think it's a rich set of 5 lessons, about an hour long each, that turn you into someone who really understands the fascial tissues and is able to better distinguish between the misinformation and the actual facts out there on this particularly popular topic at the moment. Other than that, thank you for joining me here in this lesson on densification. I really hope you learned something. Again, reach out with anything, any questions at all, and I hope to see you again soon somewhere in this sphere of learning about the body.